free to interrupt, Nikita.
All right, so hi everyone. I will be talking today, as Dennis mentioned, about the studies of hereditary cancer. And this is one part of the larger project about the pan-cancer study, which should be a part of another paper, hopefully soon submitted. And as Dennis mentioned, the traditional ways of studying studying cancer disorders for many years were mostly looking on the somatic mutations, because this is provides the most straightforward answer to what exactly causes the tumor growth. You see a mutation in a tumor, you don't see it in a normal tissue, then this is most likely the cancer-driven genetic effect. However, there is also, cancer is a type of disorder where you have both certain genetic predisposition and also the a substantial effect of the environment. So in earlier days, years ago, in the early days of genetics, the linkage disequilibrium studies were one of the kind of key methods for studying genetic predisposition. And in this case, you're kind of bound to the fact that you will be looking for the rare, extremely rare, most likely, private to a family mutations that would cause, large effects, and, it will be almost fully penetrant variants. But this doesn't provide really information about, why population-wise people, genetically have predisposition to cancer in particular. On the other side, with introduction of the GWAS methods, this kind of provides the insight into the populational frequencies of cancer predisposition. However, this is bound to common variants and presumably with a small effects. So each of those methods has a significant limitation, whether it's limited to the finding a large kindreds, which is quite rare to encounter in the clinic, or it is limited to the small effect variants. So with the introduction of the sequencing, and in particular exome sequencing, and drop of the price of the sequencing, we're now able to study rare variation in the population, which is only limited by the cost of sequencing, technically now. So this project originated from 2012, 2013, when this was still in the early days of exome sequencing case control studies. So basically, our motivation for the study was to develop a specific pipeline which would introduce a maybe not unified but somewhat standardized approach for rare variant association tests. Well, it's well known how to perform GWAS test, right, and how the whole planning of the study should be organized. It was at that time it was less known about the rare variant studies. So there was no established pipeline for the analysis of the large data set. There was no case control really studies for melanoma except occasional GWAS studies, and majority of the so far known genes, risk genes for melanoma, were discovered either in as a co-segregating variants within the families, or as a GWAS hits. And the last but not least is an interesting thing, that melanoma in particular appears to in the specific cell type in melanocytes, which are not only found in skin but also in the eyes, right? There is a possibility to have an ocular melanoma. And I think many of you heard about the specific drug introduced, I think, a year and a half or 2 years ago, which does it really well with the even late stages of cutaneous melanoma. However, nothing is really known about the ocular melanoma, and that's really aggressive cancer and highly metastatic. So so far, it was just mostly surgical and traditional chemotherapy studies, but it's a very rare cancer. So the cohort that we have in the study is about 100 patients, and it's the largest cohort of ocular melanoma, as far as I know, assembled up to date. So, okay, so basically the study design that we proposed, so all of the cases were collected from 3 different centers. So one of them is in Greece, in Athens, and thanks to Alexandros Strategos, who led this project at the Greek side. So interesting part here is that in a Southern European population, the prevalence of cutaneous melanoma is actually very low. So the frequency is very low, and actually very similar to the one observed in African American population. So this is basically a a population which is genetically protected against cutaneous melanoma, meaning they have darker skin, darker skin phototype, dark hair, and so on. So there is a genetic background that is protecting them from cutaneous melanoma. So the other cohort was assembled here by Hansen Sao at MGH in Boston. And the ocular melanoma cohort was also assembled by Ivana Kim here at Massachusetts I&D Infirmary, also in Boston. So this is a cohort of, both cohorts are considered to be genetically enriched melanoma cases. All of them have substantially early onset of the disorder. However, that was not used as an inclusion criteria. So all of the cases included in a study had at least one relative, at least one first-degree relative, also with the observed phenotype, and those are families of primary cutaneous melanoma or multiple primary melanoma. Very few of them had cases of cutaneous and ocular melanoma in a family. And the kind of most, from the genetic and statistical standpoint, most interesting about this is that we had a substantial large amount of controls and also samples that were available just for joint cloning. So we processed everything as a single batch from, all of that was sequenced here at the Broad Institute, and all of the samples were jointly processed through the alignment and joint cloning pipeline with GATK. And as a final data set, so we matched roughly about 7,500 controls, both in populational and genetic background, to cases. And that was kind of the study design. And now I want to dive into the case control matching procedure, which is essential for case control studies of the such a heterogeneous data sets, since control data set was assembled from the samples available at the Broad Institute, and those include samples from ESP study, 1,000 genomes, some control samples from other studies that were performed at the Broad Institute. So it's a heterogeneous data set, and as you can see, it's multinational. There is all kind of populations present there, and European cluster. So the samples identified as Europeans from PCA also have some clustering. Those most likely are the Ashkenazi populations, somewhat outlined from the European cluster. And what is important is that we need to introduce the batching of the European cluster. I'll show why specifically that happens in the next slide. So we just performed, identification of the several modes of the distribution and principal components, and identify the specific batches within the PCA space. And if we look on the common synonymous mutations and just perform the Fisher's exact test, ideally, we should see a completely null QQ plot because synonymous mutations are thought to have no effect in proliferation of melanoma. But if we treat everything as a single batch, so if we don't batch it at all, then we clearly see an inflation. If we stratify on the batches identified here, what this means is that now whenever I assess the p-value associated with a specific variant, so how that is usually done, you randomly permute labels of the cases and controls and see how the prevalence of the alternative allele in what is now called, after permutation, what is now meant to be cases, how strongly it differs from the actual situation, and you perform that only within a batch whenever you run a stratified analysis. So it means that you permute the labels only within the green batch separately, blue batch separately, orange batch separately, and assemble the results under the single statistics. That looks slightly better than non-stratified analysis, but still inflated. So if we look within each individual batch, then we see that one of them, and that's the smallest one, the red one over here, shows deflation and messes up everything. So we just removed it. And after all, that looks quite neat. So the stratified analysis returns a null QQ plot. That's what is expected, and thus kind of convinces us that everything is well matched, and we can proceed with the further statistical analysis. So originally, you would look at something what is already known just to prove to yourself that there are no significant perturbations within the data, and you find actually the known genes which are previously reported. And within the cutaneous melanoma, so we first confirmed several of the CDK and 2A mutations with Sanger sequencing, just to prove the point that we're doing quite good with the exome sequencing quality. And what is highlighted in red here is that recently, by now it's probably not as recently, so several years ago, POT1, so it's a gene which is a part of the telomerase protein complex. So it was identified within the family using the co-segregation, and there was a specific missense variant identified within the family that was later on functionally followed up and proven to have the causative effect to the proliferation of cutaneous melanoma. So far, no loss of function mutations were reported within this gene in the cutaneous or ocular melanoma. So what we would find here is that there are two loss of function mutations, stop gaining and the splice, essential splice site mutations, never seen in controls, but observed in our data set, strengthening the point of the earlier paper about the POT1 being a truly germline risk gene for cutaneous melanoma. So next thing we kind of out of interest, we looked at the MC1R redhead variants. So those are the common variants which are which were originally discovered by the GWAS studies, and originally people carrying homozygous mutations in the redhead variants will have red hair, and those people have a very specific skin phototype which does not allow them to tan at all. So they have the sunburns immediately, they don't really tan, and thus they're more susceptible for all kind of the somatic UV exposure driven mutations leading to proliferation of skin malignancies. So interestingly, we're getting to the point of the, that the size of the sequencing data sets are actually allowing us to capture the signal from the common mutations, which was, earlier that was just a, benefit of the genome-wide association studies. So we observe specific variant associations, and also the, if we count the cumulative count of the alternative alleles within this kind of red hair variants within MC1R, we also see a very significant association both in this genetically enriched cohort, and we also tested the TCGA cutaneous melanoma cohort, which is thought to be sporadic, or at least not ascertained for the genetic background. So further, we dived into rare variant association study, and preliminary to that, in our earlier publication, we developed a composite approach for rare variant associations, which consists of three individual models of disease that we use to assess the statistics for association. So we don't know, from scratch. So our null assumption is that we don't know what exactly type of variation would be contributing the most to the germline risk of disorder. So we test separately rare protein truncating variants burden using just a cumulative counts of the alternative alleles in cases and controls on the protein truncating variants, and just estimating the Fisher test statistic. And then we introduced also the rare missense mutations added to the rare protein truncating variants, and looked for specific patterns of variation using variable threshold and C-alpha test. So just intuitively thinking, if you just compare the count of the alternative alleles, then you can get a situation when one particular position is recurrently mutated in cases, and a different position is recurrently mutated in controls. If you just compare the alternative counts, then you will most likely see no signal of association. But the fact that two different mutations are recurrently observed in cases and controls actually brings very valuable information that is actually non-random. So tests like VT and C-alpha are actually providing information about this kind of scenarios, which is mostly observed in the missense mutations. And then the third model that we used is that there is a current quite reasonable belief that the lower is the frequency of the variant, the higher should be the size of effect that it carries with respect to phenotype. And what we did, we filtered out all of the variants observed in our data set so that we include only variants that are either not in exact database, which is the large frequency reference database of like 60,000 samples, or it's extremely rare and exact, so say less than 1 in 10,000 samples. So with the preliminary steps that we discussed earlier, so the quality check of the genotypes, the assessment of the differential missingness of the, due to coverage issues of a specific variance between cases and controls, and we also removed all of the related samples so that we kind of exclude the confounding effect. That was the our final preparations for the case control study, and we kind of came up with this nice visualization where we can use the Manhattan plots from originating from GWAS studies, but now each point on this plot is a gene. And out of the 3 models that we used for this kind of composite association test, we have chosen the minimal p-value for each gene. and respectively adjusted our null hypothesis. So if you think about this in terms of the QQ plot, then for a single test approach, you would have the observed statistics, right? The observed p-values and the expected p-values, which were generated from just a uniform distribution between 0 and 1. So since we choose the minimal p-value out of the 3 models, then we performed exactly the same procedure to assess the expected p-values. So we randomly drawn 3 uniform distributions and chosen the smallest p-value out of the randomly drawn uniform distributions for each gene entity. So thus we're being kind of conservative on the statistical approach. So on this plot, you can see just noise, obviously. There is only one genome-wide significant hit that we observed is the well-known CDKN2A gene for cutaneous melanoma, which is, both has been observed in families and also in a somatic field, also mutated. And there is some gray zone of, potential candidates, which we, just artificially limited to be below p-value of 10 to the negative 4. And the same for the ocular melanoma, where BAP1 so far is the only, as far as I know. No, there are actually three genes that are known to be somatically mutated in ocular melanoma. Yet less is known about the germline predisposition just because it's so rare. So assembling of the large kindreds of ocular melanoma is actually so far has not been done just because they're not available at all. And but BAP1 makes a perfect sense. This is the tumor suppressor gene, and it's not surprising to find it as a risk germline gene for ocular melanoma. So as one of our points and motivation for this study was to look whether there is any kind of genes that are predisposing to both cutaneous and ocular melanoma. So we generated this kind of plot to see whether along y-axis you will see the p-values for ocular melanoma, and for along x-axis you will see the p-values for the cutaneous melanoma. And while there are substantial candidates for one or the other phenotype, there's no really genes that would contribute to significantly to both phenotypes, suggesting that well, this candidate LCE1E, I don't know, is probably the only one statistically making sense, but functionally, nothing really is known about this gene, and it doesn't really look as a promising candidate for any of the cutaneous or ocular melanoma. So far, it looks like the onset of the cutaneous or uveal melanoma, whether it will be a skin malignancy or the ocular malignancy, actually has a quite substantially different genetic background. So all of the candidates, all of the candidate genes identified on those Manhattan plots that I showed you earlier were subjected for the independent replication. And for that, we assembled also a jointly processed, so it was a part of the original data set, original jointly processed. And so the primary cohort and the replication cohort were part of the same data set. So we had like roughly about 400 TCGA cutaneous melanoma cases, and extra 3.5 or so thousand controls matched to the replication case cohort. So the only 2 genes in cutaneous melanoma that were replicated was the well-known CDKN2A gene and the EBF3, the B cell factor, replicated in cutaneous melanoma, and we did not really replicate the BAP1 in ocular melanoma. However, the size of the cohort is quite small, so we cannot really prove the fact that it's germline risk gene, but functionally BAP1 makes perfect sense. And since the counts of the EBF3, so EBF3 appears to be a novel candidate gene, and the counts are quite small of the alternative cases, we also reached out to David Adams' group in Sanger and asked them for the familial cohort that they had to see whether there is also EBF3 carriers, and there appears some. We used the, what makes the perfect sense as the ancestorial consortium that was not jointly processed in terms of the variant calling and alignment and other things. So the control set for this was used as a UK10K subset of that study, which was originated from UK or Australia, so that this kind of perfectly makes sense in terms of the ancestorial background. And the joint statistics assessed using the Mendel-Heinzel chi-square test actually then brings the EBF3 up to genome-wide significance scale, suggesting that there is a substantial statistical evidence that EBF3 is indeed a germline risk predisposing gene for cutaneous melanoma. Are there any questions so far? No, not really. Can people really hear me?
Yes.
Okay. Okay, so that kind of the first part of the study, mostly statistical, right? So where there are a lot of the well-conducted studies of looking for the genetic association, both in GWAS and the sequencing studies. Not all of them, unfortunately, succeed to find the new candidates and then well replicated. So we're just it's to some extent it's also a quite lucky situation that we found this heat which was very nicely replicated and then confirmed with the second replication. So the obvious follow-up to this would be to look into the literature and see whether somebody found that EBF3 is in any way relevant to cancer. And we happened to find earlier evidence that it was involved somatically, associated with some worse outcome in the colon cancer, as far as I remember, but there was no any kind of association signal observed. So we needed a functional proof that EBF3 does something with the tumors. So what happened is we reached out also to our collaborators in Sweden, in Goran Johnson lab, and what they found is that if you look on the known somatic drivers of cutaneous melanoma, then EBF3 has also these substantial number of deletions somatically observed in a tumor, which is quite different from the rest of the genes that are not reported in melanoma studies. And interestingly, that tumors with the high expression of so people who have tumors with the high expression of EBF3 tend to do better, so they survive better with melanoma. And the other interesting point is that EBF3 appears on the same arm of the chromosome 10 as P10, which is, there is a certain evidence of the concurrent shallow deletions. However, it looks like this is mostly happening due to the deletions of the whole 10q arm, where the expression of the EBF3 and P10 does not appear to be correlated at all. So we concluded that apparently, while residing on the same, on the same arm of the chromosome, they drive some independent signal somatically. And further, we looked on the expression expression of the EBF3 within the melanoma tumor cell, so melanoma tumor cell lines of the different kind of most common melanoma types. So highly immune tumors, high MITF expressing tumor, highly proliferative melanoma. We observed that the lowest expression of the EBF3 is observed in the melanoma cell lines with the high expression of EBF3. So what then was done in both lab of Hans and Sau here at MGH and in Sweden, we looked at the known MITF targets, so tyrosinase and MLANA. So here's the correlation of expression of these guys with EBF3 and MITF, and also on the known upstream regulators of MITF, TCF4, SOX10, and PAX3. So what we observe is that TCF4 is actually a known negative regulator of MITF. It's positively correlated with EBF3, and expectedly, as I mentioned, it's negatively correlated to MITF. So what this means is that the hypothetically EBF3, high expression of EBF3 suppresses the expression of MITF, which is indeed a cancer-driving gene. So if this is true, then EBF3 should have a tumor suppressor gene effect. So this could be, this could be tested in a way that you overexpress EBF3 in the melanoma cell lines, and if this hypothesis is true and EBF3 is indeed the tumor suppressor gene, then you should see smaller tumors in the if the EBF3 is highly expressed. And this is indeed true. So this kind of cell line was infused in mice, and these are the real tumors removed from mice after the 10 days. What you see is that the cell lines infused in mice which do not express EBF3, so they have a normal levels of expression of EBF3, grow much larger than the tumors developed from the cell lines with overexpression of EBF3, thus confirming the hypothesis of the indeed tumor suppressor effect. So with that, I'll acknowledge all of my colleagues who participated in this and conclude with the with the story of EBF3, which were to some extent this is just a very this is the best kind of obstacles which all came along together so that it's both observed, associated, replicated, and very very robustly confirmed as the functional role, and also providing the specific insight into mechanism of how exactly this happens due to regulation of the MITF. So I don't know if you have any questions, I'll be happy to take them, and let me know if anything in particular from the statistical standpoint, how the data set was QC'd. You want to hear more of that, since this is kind of the most important probably part of this analytical approach.
Yeah, thank you, Mykyta. This is very interesting and very. I think you really did the most of it for the small cohort, right? So that you get up into a very good candidate which you could replicate and even functionally prove at the end. So are there any questions? Keep in mind that you're all on mute. You have to unmute yourself.
All right, so I'll then just unravel some